So my name is Ciara Finucane, and I'm the Senior Director for Contract Research Europe. So I'm based in the Invicro London office in the UK. And I'm delighted to introduce Dr. Kavitha Sanasi, who's been one of our early users of VivaQuant, even when it was back in the InVivoScope days. So I've been using it for many years. And she comes from the Division of Imaging Sciences and Biomedical Engineering from King's College London. And she's a preclinical imaging facilities manager there. And she's going to talk about PET imaging, looking at changes in copper metabolism in an Alzheimer's disease model, and evaluating the VivaQuant brain atlas segmentation with an in-house texture analysis. So thank you very much, Kavitha.
Hello, thank you. And thank you for inviting me to be a speaker at the user meeting. It's my first visit to Boston and to Invicro, so thank you for making me very welcome. As Ciara said, I'd like to talk to you about the work that was done by these authors, detailed below, but it was mainly Julia Torres. It was her PhD project. So I will be describing her work during this talk. So just a little bit of background. So talking a little bit of biology. Her work was she was interested in Alzheimer's disease, and as you know, it is a fatal neurodegenerative disorder. And as you may know, there is no conclusive way of diagnosing it at the moment, other than at the post-mortem stage, that you can actually say definitively, yes, that patient had Alzheimer's. So in a way, it's a holy grail to try and figure out if we can do diagnosis, but especially early diagnosis, and preferentially in a non-invasive manner. The disease is characterized by neuronal loss, neurotransmitter imbalance, neuroinflammation, brain deposition of neurofibrillary tangles and FTs, and also amyloid beta plaques, which are very characteristic of this disease. So because we don't have a reliable and objective diagnostic marker, the aim of this study was to try and look at a different aspect of the disease to come up with some sort of PET marker. We were interested in copper because you may not know this, but copper is the third most abundant trace metal in the human body after iron and zinc. You would obtain your copper through your diet. Copper actually serves as an essential catalytic and structural cofactor in a number of important enzyme-mediated processes. And the copper homeostasis in the body is actually not very well understood and still under investigation, but there is a balance between its essentiality and also its toxicity. So copper metabolism imbalance is known to be a key pathological hallmark in a number of human diseases, including Alzheimer's disease. The brain is actually one of the most copper-demanding organs in the body, bar the liver. And we're able to work with the isotope copper 64. So we were interested in following copper in the brain because we know that it's involved with many of the landmarks of Alzheimer's disease, such as amyloidosis. So we know that copper will bind to the structures that are formed when the amyloid beta plaques are formed in the brain. It's involved in synaptic toxicity. We know it's also involved in oxidative stress, as it behaves as a cofactor for copper-zinc superoxide dismutases, so to control the oxidative stress in the brain. So we know that brain copper dyshomeostasis is involved in Alzheimer's, and the study was to look at that in more detail. So we used copper 64 trafficking and monitored it by PET in an Alzheimer's mouse model called the TAS-TPM model, and we know that this particular model will exhibit Alzheimer's like amyloid beta plaques. And we also use the tracer copper 64 GTSM as our probe. The other aim was to map the distribution of copper 64 in the brain, looking using VivaQuant, using the atlas-based analysis, using a predefined brain structural template, and comparing it to texture analysis. And we used an in-house MATLAB-based program, which was which focuses on the study of the spatial interrelationships of the image voxels and their intensities. So our methodology. So the contrast agent we used was copper-64 GTSM. This particular molecule has moderate lipophilicity, and it is capable of crossing the blood-brain barrier both in wild-type mice and in the diseased mice. It's also prone to bioreduction and the metal release in a non-specific manner. So it will enter the cell and as copper-2 GTSM, and then it will turn into copper-1 GTSM, or the copper alone, and enter the pool of intracellular copper. And so this is what we were following during our PET imaging experiments. The model we used was a TAS TPM mouse model, and we obtained this model through our work with GSK. It's that particular model. These mice are known to express amyloid beta plaque deposition, is known to exhibit neuroinflammation, and our colleagues at King's also have data showing you have age-dependent cognitive decline at around 6 months. So when the mouse is 6 months old, their behavior starts to change, and so we were interested in monitoring copper around that age in their brains. The imaging protocol is as follows. So we injected the mice intravenously with copper-64 GTSM, about 35 megabecs, and we did an immediate scan on our PET-CT, which is Imedito PET-CT system. And we imaged from 0 to 30 minutes, and then we allowed the mice to recover, and we re-imaged 24 hours later, and then we culled mice. And we did either, obviously, image reconstruction and the PET ROI analysis. But we also, so the ROI analysis, we looked at the brain atlas quantification using VivaQuant, and we also looked at texture analysis. We also did some other analysis methods, such as ex vivo brain autoradiography, some histology, and some biodistribution. So the results. I don't know if those of you who are aware that VivaQuant have a Atlas plugin, and it is either mouse or rat, and so we use the mouse version of the Atlas, and it can give you brain-defined areas within the Atlas. So what we did was the brain ROI was drawn in the CT image, then it was applied to the PET image of the TAS TPM mouse and the control mouse. The atlas was first scaled and reorientated on the CT image, and then the PET image was then overlaid for ROI segmentation. What we found was, as you can see, the TAS TPM mouse brain compared to the wild-type brain, just by visual observation, you can see that the hotspots, they do look different, and these are ex vivo brain mice at 24 hours. In the middle, these are autoradiographs of the mice brains, and again, just visually, you can see there is difference. So the other confirmation of this difference was that these autoradiograph slices were then, the same slice was then used in histology stained with Congo red to label the amyloid plaques. And as an aside, what was interesting, the hotspots where the copper was found, particularly in the TAS TPM mice that would have this amyloid load, did not co-register. So that was quite interesting in terms of the development and the changes in the disease. So the two didn't co-register. So here, looking at the data, what we found was that in figure A, in the histogram, what we could see was that the diseased animal, the TAS TPM Alzheimer's mouse, showed significantly higher concentrations of copper-64 in the brain at 30 minutes, and again at 24 hours. What's also interesting was that we know that this is free copper in the brain and not the copper GTSM species, because some mouse brains were processed and used in a solvent extraction process to identify the species of copper, and there was no intact copper GTSM. So what we're looking at is free copper that is now residing in the brain. And what is interesting is in the wild type, you can see that the amount of copper that goes in at 30 minutes seems to remain stable after 24 hours. In the figure B, what we're looking at is the clearance rate of copper over time. So the blue lines show you the wild type copper, which seems to remain stable, and the TAS-TPM mice, between the 30 minutes and the 24 hours, you seem to have a faster clearance rate. What's interesting is that even though there's a faster clearance rate, because the uptake is already so much higher, even though you have a faster clearance rate, you still have more resident copper in the brain of the diseased animals. So using the VivaQuant atlas, which I described before, if you overlay those areas that are already identified within the atlas onto the brains of both our TAS-TPM animals, which are the red boxes, and the wild type, so at 30 minutes, well, the first overall impression you would get is that the copper uptake in the various regions of the brain in the diseased animal is generally higher than in the wild type normal controlled age-matched animals. And in some cases, there are significant differences in the uptake in particular regions of the brain. If you look at the 24-hour time point, again, you see that overall, the normal level of copper in a normal brain stays relatively stable compared to the 30 minutes and 24. And again, the TAS-TPM, this diseased animals, still show enhanced copper uptake in the brain and very specifically localized to very specific regions of the brain. So there is a statistically significant difference in regional copper brain concentration. So the regional distribution of the tracer is at or beyond. This is the issue that we had and why we decided to use a different method in a way to almost complement the atlas quantification and to give us some reassurance regarding the data that we're seeing. So one thing is that the regional distribution of the tracer is at or beyond the resolution limits of the PET-CT scanner, because we're talking about the brain, and this is a very small structure, a mouse brain. And also, we realized that the heterogeneity in the distribution of the copper-64 in the brain was probably underestimated in terms of quantification by the atlas-based method due to partial volume effect. So we're talking small structure and also smaller substructures and partial volume effect combined. So we decided to use a different analysis method called texture analysis. And texture analysis, for those of you who don't know, is a mathematical method that is used to describe the relationship between the gray level intensities of the pixels or voxels and their position within an image. So we're now interrogating the voxel intensities within an image. So this particular method is based on a statistical method, and what you're looking at in a way is not really related to anatomy and it's not related to biology, but you're just looking at two populations and you're asking the question, how different is one to the other? And is that a real difference and is that significant? And within those differences, there are various parameters that can be used to describe the differences in frequency distribution of your voxels within your ROI. And these are not, on this particular method is based on a histogram analysis. So the way this was done was the PET and the CT images were imported into our MATLAB-based in-house program, the region of interest was drawn on the CT because it's obvious the brain is very easy to define, and B shows you what the ROI would look like, and C would show you the histogram of uptake. So much of the data is looking at the differences in the histograms between a normal population and a disease population. So as a biologist, I had to go and try and understand some of the language that's used to describe these differences within the populations. Some of the terms will be familiar to you. So we're looking at first order parameters. So we're looking at 1 pixel statistics. So mean, min, maximum, median, percentile, standard deviation. These are probably familiar to you, but skewedness, kurtosis, coefficient of variation, energy, and entropy might not be so familiar. So skewedness is a way of measuring the asymmetry and the deviation from a normal distribution. So if you imagine a normal distribution on a curve, the skewedness, if it's perfect, it would be 0. And so the distribution of those voxel intensities would be symmetrical around the mean. Kurtosis describes voxel intensity cluster around the mean. So if you imagine your normal distribution, a perfect normal distribution, you would have a kurtosis of about 3. A lower kurtosis number indicates that there's less cluster around your mean, and therefore there's more spread of your voxel intensities away from your mean. Coefficient of variation is a measure of relative variability. So it is the ratio of the standard deviation to the mean. So the take-home message is the higher the CV, the more variability relative to the mean is what you're seeing. Energy is a measure of uniformity in your population in terms of your voxel intensity. So the way I would imagine this is if your normal distribution was a straight line, so everything was the same, you would have the highest energy. But conversely, you would have a low entropy because everything is the same. So you would have low entropy. So entropy is describing the spatial randomness within your population. So it's actually characterizing the heterogeneity of the sample. So I know this is a lot to take in, but and this is kind of what you end up with. If you use a program, you end up with all this data and all this information. So it's trying to understand what that means and interpret it. And this is probably the more simple version of texture analysis. But if you look at the pink bars, for example, if you look at the TAS-TPM group of mice and you compare it to the wild-type mice, so your kurtosis for the wild-type mice is around 3.0 at 30 minutes, and it seems to stay quite similar to 24 hours. And if you remember, I said a kurtosis of about 3 gives you a nice normal, is closest to a normal distribution. And a kurtosis of 2.97 and 2.78 is telling you, here you go, kurtosis is that you have less cluster around the mean and you have more spread. So, for example, if you look at entropy, for the wild-type animals, you have an entropy of 1.39, 1.48 between 30 minutes and 24 hours. But in your sample of your diseased animals, your entropy is higher, which suggests more heterogeneity in your population. And conversely, energy is lower in the diseased animals compared to the wild type. So the take-home message is that the statistical analysis reveals a higher spread, asymmetry, and non-uniformity of the voxel intensity distribution in the TAS-TPM brain population, indicating a greater degree of image heterogeneity in the TAS-TPM disease model compared to the wild-type controls. So to summarize, with the copper-64 GTSM PET imaging, we were able to demonstrate that there was a significantly increased brain concentration of copper-64 uptake at 30 minutes and at 24 hours post-injection of the tracer in our TAS-TPM animals, and also faster clearance of the copper from the brain in the disease model compared to the control. The Atlas-based PET quantification and the texture analysis both demonstrated substantial variations in the regional brain concentration of copper-64 and PET image heterogeneity, respectively, between the two groups. So both were in agreement with each other, but in different ways. The study provides an initial set of PET image parameters, so looking at global brain concentration of copper-64, rate of clearance of copper-64, degree of heterogeneity of the brain copper-64 distribution, that we could potentially evaluate in early human trials to determine the potential of brain copper imaging and trafficking as an early diagnostic marker of changes in the Alzheimer's disease brain. So finally, thank you for your attention.
I was just wondering what the liver uptake looked like over time in the disease versus the control.
We did do. So the question is, what did the liver uptake look like in the disease versus the control? So we did do it. So we did do the whole body biodistributions and imaging. I'm just trying to remember. I think we had high uptake initially in the first 30 minutes. I can't quite remember how the washout was after 24 hours, but I think there was quite high washout in wild type. And I think the only organ of significance that seemed to retain copper at 24 hours in the disease animal was lung, which was unusual. So the washout in both disease and wild type was quite similar. They both came down because obviously 30 minutes is very quick after the injection. But lung was the only one that had a significant difference in that there was higher copper in the diseased animal, and we don't really know why that is.
The liver uptake in both disease and wild type was similar throughout, looking at 30 minutes and 24, or was there that's what I'm the brain to liver ratio.
Yes, you're right, because copper, liver. I'll have a look at that and whether we can compare the two ratios between liver and copper. But I can show you the data for the whole body and just verify that for you.
Thanks for a great talk.
Oh, thank you.
I think we had a couple more questions.
Have you found like in my even control mice was some agent like multiple sclerosis or some do you find a big difference?
So if we give the mice what agent? Ah. Well, that's the next, obviously the next step. So the question was, have we fed our animals or given them a copper chelating agent? So what is known in the literature is that if you give a copper chelating agent, and there are two, there's cleoquinol and PBT2, they tend to strip the amyloid plaques of copper, and we think that it allows redistribution of copper in the cells, so you then, in the mice, recover cognitive function. We haven't done this yet, but I have a collaborator who wants to do exactly that very shortly. So we will then image and see. But I think his interest might be using, instead of copper-64, will be to use some of the PET agents that we use in human patients to label amyloid plaques. So you have the F18 fluor beta-peer, fluor beta-ben, or flubetamol. So we're a little bit dependent on when a clinical trial is happening on a patient and our PET center is making that particular compound, and then we would use that to evaluate the amyloid plaque load first, and then look at chelating effects by stripping down. Yeah, see what happens. Thank you.
Have you performed your texture analysis on individual brain regions of interest? So I think what you showed was whole brain.
Yes, so I think for the purpose of the PhD work, it was done on a global brain, partly because of the reason of that if you're trying to look at small areas, you have partial volume effects, so the whole brain was taken as a whole. But of course, there's nothing that would preclude you trying to do very specific ROI areas and then interrogating that using the texture analysis. And as you may know, there are more interesting or complicated ways of doing texture analysis, so not just looking at one voxel, but the relationship of one voxel to another voxel, their proximity and their similarity, the frequency of intensity between two voxels, also the angle at which the other voxels are at relative to the voxel you're interested in, and also there are other ways to interrogate the data to look at run lengths of voxels. So if you had a homogeneous area in an image, you would expect the voxels to have similar intensity, so you would have a long run length of that intensity, whereas if you had very heterogeneous sites in your image, you would have shorter run lengths. And so there are other kinds of matrices that you can use to interrogate your data. This one was done with just first order parameters, but we could remine the data and look in more detail. But I do know that this work, this particular software is also being used to look at tumor models in mice, so comparing vivaquants in tumored mice versus texture analysis, but it hasn't been published yet. But originally, the software was developed to look at clinical images by the authors who are actually clinicians, and they want to look at better ways of evaluating FDG uptake in a tumor. And so it's in a way we're going backwards from the clinical to preclinical.
Great. Time for one more question.
Wondering, have you ever done a control experiment where you're just injecting free radiolabeled copper?
Yes, we have.
You just set out that baseline.
Yes, so that's all been done, and that's just, I think the paper is just coming out now, and it's looking at where just copper acetate just goes as a salt, where it goes in the brain, and also the washout and the flow in normal brain versus diseased brain. So yes, that's been done. I'll give you the reference within the paper. It's about to be published.
Fantastic. Okay.
Thank you very much.
Thank you very much to Kavitha again for an excellent talk.